Hello everyone, Dr. David Perlmutter here. Welcome again to the Empowering Neurologist. We have a really interesting program today, and I just realized, I think I might say that every time. And what can I say? We have interesting guests. Today's guest is no different. Dr. George Tetz is doing some amazing work looking at what are called bacteriophages. And, we've talked about viruses, we've talked about bacteria, we've talked about protozoa, all kinds of groups of organisms that are involved in the human microbiome, but we haven't really talked about phages. And phages are a form of virus, but they are specific in that they infect bacteria, so we call them bacteriophages. And it's not like this is necessarily something we haven't known about. Bacteriophages were first actually described in 1917. The former Soviet Union manipulated bacteriophages to use as antibiotics decades ago, and yet, for whatever reason, they really have not been the subject of intensive research. Well, that's changing now, and I think what we're going to learn today, according to some recent publications authored by our guest, Dr. George Tetz, that there are some implications of these organisms, bacteriophages, in terms of influencing bacterial activity and how bacteria are able to do the things that we want them to do, for example, maintain the gut lining, and how these may be influenced by being infected, if you will, by bacteriophages. So his work is getting a lot of attention lately as it relates to neurodegenerative conditions, things like Alzheimer's disease, Parkinson's disease, autism, not specifically a neurodegenerative condition, and even type 1 diabetes. And I think that as we work through our interview today, we may well be able to extend the possibility, at least, of this mechanism to other autoimmune conditions. So we're going to talk about some of his recent publications, some of his new research, and I'm really looking forward to our interview, so let's get started. So Dr. George Tetz, it's an honor to speak with you today. Very excited to hear about your research.
Hello, good morning. Nice to speak with you too.
As well. Okay, so first of all, maybe you can tell us about your laboratory and what you're working on right now.
Sure, absolutely. So I'm the co-founder of Human Microbiology Institute. That is a multidisciplinary microbiome-focused company. And we're located in New York. Originally had a lab space in Boston and Lexington, but right now doing the science in Big Apple. So actually, here at the Human Microbiology Institute, I have absolutely come to believe that we cannot understand human microbiome and microbiology without looking only at the bacteria within human gut. So we're understanding a new world of microbiology where not only bacteria, but also bacterial viruses, named bacteriophages, affect human health. And actually our core was the first to suggest that bacterial viruses might be previously overlooked human pathogens.
Well, I wanted to, for our viewers, you're all very much familiar with the exploding research that's dealing with the microbial changes that are happening in the human gut and how that is manifested in disease elsewhere in the body, including the brain. And, truthfully, as you well said, we are focused primarily on bacteria. But your work indicates that these phages, these viral particles, are highly influential or potentially highly influential in the function of bacteria and therefore might well be playing a role in some of our most dreaded conditions.
Yes, I absolutely agree with you, because bacteriophages that are viruses, and for a very long time they were considered not to affect eukaryotic cells and to interplay only with bacterial one. And actually that is why for many decades of microbiome research, no one paid real attention to them as possible pathogens.
Well, and even more specifically, mammalian cells. But we now, through your work, are recognizing that these phages do, in fact, play a role. And this isn't necessarily new science, is it?
I absolutely agree with you because actually our discovery and the ongoing discoveries, we have not stopped on already published works. So actually, we're showing that phages are previously overlooked triggers for both very early diagnosis of pathologies as well as possible targets for the therapeutic interventions, meaning that to use phages as a novel therapeutic targets.
So again, this is a very important point for our viewers, and that is, while it's great to talk to Dr. Tetz about all his work and how exciting this research is, the play here is that this might well contribute to some actual therapeutic interventions for our challenging issues with Alzheimer's and Parkinson's, for example. One of your papers, I have it here, Parkinson's disease and bacteriophages as its overlooked contributors. So I know in that study you characterized the bacteriophage array in Parkinson's patients versus those who did not have the disease, and you found some remarkable differences. Can you unpack that for us and walk us through what your findings were?
Sure, absolutely. So with respect to the Parkinson's disease, we demonstrated that bacteriophages are intrinsically involved in particular alterations of the microbiome that in turn are implicated as a triggering of Parkinson's disease. So just to highlight that in this paper we have shown that phages are the original factors that trigger microbiome reshaping it in a pathological condition. So our attention was particularly drawn to a decrease of Lactococcus bacteria, Lactococcus species, in the Parkinson's disease group, and the fact that the decrease was due to the increase of lytic lactococcal bacteriophages. And it's important because the decrease of Lactococcus species, it is a very important triggering factor for different neurodegenerative diseases, including Parkinson's. And the association of the decrease of these bacteria with the alterations of enteric nervous system is pretty well shown in multiple and different articles that have shown this association.
So the literature shows us that there are variations in the makeup of the bacterial components of the gut that are seen in Parkinson's, certainly autism, Alzheimer's as well. And but your contention is that one of the factors that may be playing a role here is not just the array of bacteria, but their functionality may be changed because of the influence of these particular phages that you have now identified.
Yes, thank you. You're absolutely right. Because actually, a lot of research, particularly microbiome research, is showing the association of the alteration of certain bacteria and the development of different diseases, or just association, or changing of metabolome profile, etc. However, the question, what leads to these alterations that are seen? And actually, this is the question that really is not answered by scientific community yet. There are certain alterations for the antibiotic exposure that is definitely an important source of the alterations of microbiome. However, the alterations of the phagobiome, and this is the new term that we have introduced to describe the totality of bacteriophages population, is even more influential and is even more important. Because there is a great article named Movers and Shakers about bacteriophages that actually show that how did bacteriophage can change in a microbiota community by the alterations of their population, of their viral population.
One of the mainstream reports on your work got a lot of traction on the headlines because it said that you believed, or that research shows, which was your research, that there may be that neurodegenerative conditions may be contagious based upon the bacteriophage theory that you're exploring. How did you respond to that, and how accurate would that headline have been?
So actually, I'm a scientist, so I can claim something once I have proved it. Therefore, we have not conducted experimental work that would prove that these diseases can be contagious due to bacteriophages spreading. However, on the other hand, if we can imagine the situation that yes, certain bacteriophages cause alterations in the mammalian gut and in some susceptible hosts, in this case, it can easily be suggested that indeed these conditions can be contagious. Because, well, let's take a look. Bacteriophages today are the most widely spread biological entities in the world. So, for
hold
example,
right there. Please say that again.
Sure. So, as for now, bacteriophages are the most widely spread biological entities in the world. For example, just one drop of seawater, it contains 10 in 8th degree of different bacteriophages. If we'll take a look into our microbiome and to play a little bit with numbers, so we will get that the human body consists of 10 in 13-fold of eukaryotic cells. The number of bacteria associated with our microbiome is from 3 up to 5 times more. And the number of bacteriophages is 10 times more than the number of bacterial cells. In other words, the number of bacteriophages associated with our microbiota, it's eventually like 50 times more than human cells. And of course, it opens the discussion about whether or not these conditions can be contagious. However, to claim such a thing, it's necessary, of course, to add additional experimental design and much more experimental work on that.
There's a really interesting...
So I would be very accurate without these suggestions. However, if to take a look for one of the pathways for the science and for the scientific discovery, from my perspective, it seems to be very interesting.
So, whereas the bacteriophage itself does not, may I say, infect, I mean, when you look at the mechanisms by which they inject their genetic material into bacteria, it does look like an infectious type of arrangement. But that said, the party line has always been that while this is something that may happen or does happen in bacteria, hence the name bacteriophage, it isn't directly happening in our cells. But nonetheless, you've made this very dramatic connection because of the identification that you've made of the changes in the bacteria and their functionality, and therefore how that affects us as the host of the bacteria. So there is a pretty straightforward connection, although it's maybe
absolutely.
One step removed. You know, one of the things that is really hot right now is this misfolding of protein concept as it relates to various neurodegenerative conditions. That proteins, alpha-synuclein in the case of Parkinson's, for example, amyloid in the case of Alzheimer's disease, various proteins seem to be hallmarks as well as perhaps contributing to the disease itself. What role might these phages have in protein aggregation?
Sure. So actually, a few words just to add about the prions. Prions are molecules that are characterized by self-propagation, which can undergo a conformational switch leading to the creation of new prionic proteins. And actually, originally are suggested to be implicated only in two main pathologies: it's Coeur disease and the Creutzfeldt-Jacobson disease. Right now, recently, these prion proteins have been shown to contribute to a variety of different neurodegenerative diseases, such as alpha-synuclein for Parkinson's disease, TDP-43 for amyotrophic lateral sclerosis, beta-amyloid and tau protein for Alzheimer's disease. And the protein misfolding, it leads to the formation of aggregates that possess cellular toxicity, leading to neuronal alterations and the death of neurons. And we were the first to identify a variety of prion-like domains on the surface of different bacteriophages. And these prion-like domains, they share a strong capability to become prion proteins themselves. So therefore, actually right now there is an ongoing discussion about the trans-kingdom alterations of the prions between eukaryotic world and prokaryotic world. And actually in the manuscript by our colleagues Chen and Dole, they have recently reported that in their rat model and in the model of Caenorhabditis elegans, who were fed with prion protein producing E. coli, the model animals, they showed an enhanced prion aggregation in the brain, therefore suggesting such a cross-kingdom prion alterations. And if to take into consideration that we have identified bacteriophages in a wide variety of human biological fluids, starting from blood serum and including cerebrospinal fluid, the question about could prion-like proteins on the surfaces of these bacteriophages could trigger and could possess a seeding potential for the protein misfolding and formation of prions. So right now we're just finalizing another paper on that, showing the presence of certain bacteriophages in human cerebrospinal fluid, and we'll present data hopefully by the end of this year.
Getting back to the mechanism that we had discussed earlier, just the now having had that discussion just now, going back to what may be far simpler, and that is the functional changes that may be imparted into our gut bacteria as a consequence of them being infected by phages. And I think mechanistically, you've done a lot of explanation with respect to changes in endotoxin, changes in LPS. Our viewers are certainly familiar with that role in terms of gut permeability and having immune dysregulation consequences and inflammatory consequences as well. So do you think and foresee that there'll be a burgeoning of literature that connects phage involvement with bacteria with typical autoimmune conditions, whether common autoimmune conditions, type 1 diabetes, celiac disease, etc.?
Yes, and actually, you just bite my call, because we, together with our colleagues from New York University, we just have completed a very big study for the longitudinal analysis of microbiome from children with type 1 diabetes. And actually, the thing that we have identified in this study is the first microbiome-associated triggering factor for the onset of autoimmunity. And so actually, we have identified that kids who further developed seroconversion to autoantibodies or developed type 1 diabetes, they had an initial abnormally high level of certain gut bacteria. The following death of this bacterial population was totally due to the massive prophageous induction in leading to the disappearance of this bacterial population. And these bacteria, notably, it is very particular for human gut because it possesses certain pathogenic associated molecular patterns that are highly immunogenic. In other words, the situation that we have seen in this study is that these children developed autoimmunity due to prophageous induction and the release of certain immunogenic PAMs, so-called pathogenic associated molecular patterns. We're submitting this paper today.
Today, lucky us. So, you're certainly then completely on board for what some may still consider to be conjectural, and that is this relationship between microbial disruption gut permeability, and then immune dysregulation. I mean, that seems to be more and more well accepted by most, although some would still reject it. And I would say that, work by Dr. Maria Dominguez-Bello there at NYU as well really talks about prenatal and perinatal influences on the milieu of the gut bacteria as it relates to further risk down the line of autoimmune conditions.
Well, I agree with you. So, and I think it's a pretty normal situation in science when some novel scientific avenues, they should not be accepted by everyone. It would take time once some ideas would be proven, not by different laboratories throughout the world, and once it will be accepted by the medical community. Actually, I can tell you I'm honored because today, the Nobel laureates for the medicine, they have received the Nobel Prize, and actually the person who first introduced these checkpoint inhibitors pathway and suggested to use their immunotherapy in cancer, if you will talk with their oncologist 25 years ago and telling them about their immunotherapy in cancer, no one would believe that it will work out, actually.
Well, I think your statement makes me feel good. I mean, years ago when we proposed fecal transplantation for autistic children, the pushback was breathtaking. And I think that most people are aware of where that science has gone. I'd like to take our conversation to a place of your work providing the foundation ultimately for some therapeutic interventions in these challenging disorders. Alzheimer's, for example, where we have no meaningful treatment at all whatsoever. How might our understanding of phage activity lead itself to the development of some sort of intervention if, in fact, this whole downstream effect of phage infection may be operative?
Well, as I mentioned, it is crucially important for the scientists to understand and to figure out the real cause of the disease and the real cause of the problem, because it would allow to develop the real therapeutic that could target the real cause that leads to the disease triggering and progression. Therefore, we believe that our concept opens, give opportunity for principal and new interventions in both diagnostics and therapeutics. Because right now for the Parkinson's disease as well as for type 1 diabetes, so we're developing the diagnostics for very early diagnosis of the patient population who would likely further develop these conditions. So the same stuff, we are also working on the therapeutics based on bacteriophages as novel therapeutic targets for these diseases. And right now we're just launching the new studies for the patients with Alzheimer's disease and with amyotrophic lateral sclerosis in order to figure out and to evaluate the implication of phages in these disease progression.
I want my viewers to all get their arms around the notion that this is a brand new playing field. It's brand new information. I can't say that current science has exhausted the other possibilities, but for example, as it relates to Alzheimer's disease, this notion that the disease is caused by the accumulation of beta amyloid, end of story, and therefore treatments should be developed to rid the brain of beta amyloid, I think has proven to be not a good pursuit. That technology, whether it's through immunotherapy or other means of regulating or downregulating enzyme function, has really not worked. And it is, we're really looking forward to following your work in the years to come because it's very enlightening. And I think, beyond your laboratory, this is going to really provide some solid foundation for researchers around the world. So it's really very exciting.
Thank you very much. I appreciate. Thank you for kind words, and yes, we'll try our best to make new discoveries and to bring them to patients.
Great. Well, thanks for being with us today. Incredible information. I hope to see you soon.
Thank you very much. Appreciate. Thank you very much, and have a great day. Bye.
Bye-bye. Well, as promised, that was a fascinating interview. When you realize the role that these phages play in regulating bacterial function, and as he described it, the differences in the phage distribution in his recent study and how that might affect how bacteria are functioning in the human body, it really adds yet another layer, perhaps of complication, but I would call it opportunity to understand this science in terms of being able, moving forward, to possibly leverage this information and create novel therapeutic ideas, especially as it relates to things like Parkinson's and Alzheimer's, where truthfully these days we don't have a lot of options. So very interesting information. Hope you are able to watch the video link that we attached in this program to the video on YouTube about how phages work. Really very interesting. I'm Dr. David Perlmutter. Thanks for joining us. We'll see you soon. Bye for now.